All right, Dr. Tracy, come on up here. She's our star that we met on September 8th. We really appreciate all the work that you're doing and your team. And you lead the discussion now. Let's get your report.
Okay. Good morning, everybody. Today, what I would like to do is talk to you about what we've learned about the brown marmorated stink bug to date, although Congressman Bartlett did an excellent job in sort of summarizing the issues, I think. And also, I would like to talk about two key projects that we've been working on in our laboratory at the Appalachian Fruit Research Station in cooperation with colleagues at other universities, like with Dr. Chris Berg at Virginia Tech. So today we'll talk about some of our work toward development of behaviorally based monitoring tools, as well as research we've conducted toward development of management strategies for the brown marmorated stink bug. So to begin with, a little bit, just like Congressman Bartlett said, this insect has become, in this region of Maryland, a season-long threat to commercial tree fruit, as well as many other crops. It has become such a persistent problem because of the movement from outside orchards, from wild host and other cultivated crops into our orchards season-long. We also, as Congressman Bartlett mentioned, both the adults and the nymphs feed. This is fairly different from our native species. Typically, we generally would worry just about the adults rather than the adults and nymphs. The other thing we observed in 2010, which was fairly different again from our native stink bugs, is that they can reproduce in commercial orchards. So there you have a breeding population right in the orchard itself. And you can see the different stages of the different life stages of brown marmorated stink bug with the egg mass, the first instar nymphs, the seconds through the fifths, which do feed actively on fruit, and then the adult male and female bug. Now, one of the things that we learned in 2010 was that this insect, in fact, had 2 generations in our region. We learned this through some field trials at the Appalachian Fruit Research Station. So what this means is that early in the season, and last year it began early to mid-April, we started seeing overwintered adults. That's the stage that hibernates or spends the winter in a state of dormancy, starting to move into commercial orchards. Those adults began to mate and reproduce and also feed. Those eggs hatched and those nymphs developed. They developed to the adult stage by about July. So in July, those new generation adults or summer generation adults began to reproduce, feed, and mate as well. More nymphs hatched from those eggs, and then those nymphs matured to the adult stage, and we had a second full generation by late August, early September. And so what this means is really a season-long threat from all life stages of this insect, beginning mid-April till harvest. And so these are sort of the 3 major categories of problems that we saw in commercial tree fruit in 2010. First of all, we had a lot of early season injury that affected particularly our stone fruit crops, where you see the pockets of dead necrotic tissue from that early season feeding. Season-long, we saw feeding injury from brown marmorated stink bug on many different varieties of tree fruit. In this case, these are pink lady apples nearly 2 months away from harvest, and you can see the extensive injury. And then, as the Congressman mentioned, late season, just prior to harvest, we had feeding from brown marmorated stink bug. When that fruit was harvested and put into cold storage, essentially, the injury was invisible. It was not detected. The fruit was put into storage, and several weeks later, the injury began to appear. And so this was also a critical issue. So in our lab, we've been focusing on 2 key projects. The first is development of a monitoring tool, because at this point, we had no established monitoring tool for brown marmorated stink bug. But we know that the grower community needs a monitoring tool that will allow them to detect the presence, abundance, and seasonal activity of this insect, and ultimately that it can be used to develop treatment thresholds that will allow us to then determine when we need to make management decisions. And so in our laboratory, we generally study the behavior and ecology of insects, and when we design monitoring traps, we look at 4 key components. These include visual cues that the insect is attracted to that we can incorporate into a trap design, olfactory cues, in this case pheromones or other odors that the insect is attracted to as well that can be used as a lure, a capture mechanism that is compatible with their movement, with their patterns of movement, whether they walk or fly, something that works with the insect in terms of capturing them. And then the deployment strategy, where and when do you deploy these traps? And so with that, we had a number of experiments we've conducted between 2009 and 2010 that have contributed to this goal of developing a monitoring tool. The first is looking at the impact of visual stimuli, in this case, different colors that represent different stimuli to the brown marmorated stink bug. So these are the bases of our traps. So for example, black represents a trunk mimic. So if you can imagine you were a small insect foraging through the grass, looking up at this 3-foot structure, it might look like a trunk of a tree. We also have foliar mimicking colors, green and yellow, and then white and clear are more control. In other words, they're controls that they don't represent a particularly visually stimulating source at least based on our visual acuity. And so we deployed these traps at our laboratory in the field, and these were baited or unbaited. So we also evaluated a potential lure for this insect, which is a known attractant, but it is not the true pheromone of the bug. And we conducted these studies over 2 years. And what we found was, first of all, you can capture a lot of brown marmorated stink bugs with these traps. In fact, on a single day, we would capture over 2,000 adults in this experiment alone. So tremendous numbers of bugs in the environment. But in terms of the overall results, what we observed was 2 things. First of all, baited traps were much more sensitive than unbaited traps, which was good. And secondly, the dark visual stimulus of either a black or a dark green pyramid appeared to be more visually stimulating. And so with those, based on both adult and nymphal captures, we evaluated those in 2010 in commercial orchards. And we also were able to do a second piece of this sort of evaluation, in that we could also evaluate this prototype pyramid trap that we were developing against commercially available traps from Japan, where they've had the insect as a pest. And so in this case, we'd evaluated our black pyramid trap against 2 traps that allowed us to evaluate different capture mechanisms. In this case, both of these Japanese traps are deployed in the tree. Ours is deployed on the ground. And they're slightly different in terms of capture mechanism, in that the center trap is a yellow bucket trap. So it's filled with water, and the bugs must land on the trap and then fall in the pool. And then on the far left is a clear pyramid sort of style trap that's deployed in the canopy. So we evaluated these, and we found that indeed our prototype pyramid trap was significantly better. And so this is the model that is going forward for 2011 again in commercial orchards. And in fact, we are scouting for positions this week, in fact, and we were in a number of commercial orchards in Maryland and West Virginia. So these traps will be deployed between trees in the border row. They are baited with a known attractant, methyl decatrienoate. It has limitations, which I'll discuss a little bit later, but we are going to be monitoring 6 commercial orchards in Maryland and 3 in West Virginia, and using these traps as an indicator of presence, abundance, and seasonal activity. Now, we would like to improve these traps to some degree, and so one of the things we hope to do is tap into this response we've seen, perhaps some of you have seen it as well, where they respond to dusk to dawn lights, particularly in the summer. And this photograph was taken at a dusk to dawn light in West Virginia, and the aggregation of adults was estimated to be about 1 million individuals. And so that's a tremendous response. So how can we tap into that? Well, what we've done in our lab is develop what we're calling our light bioassay to define the wavelengths and intensities of light this insect responds to, and then perhaps we can incorporate them into a trap. So at the top you see, with the number 1 photograph, you can look into our light bioassay chamber. That white ring is the release point. Number 2 sort of highlights the small crescent you see in the corner where the light source is entering. This is a simple choice bioassay. We release an insect in the center, pointed away from that light source, and then we add the lid. Fortunately, we were able to create a situation where we can use a camera to follow the insect's movements and look at how they respond. And so essentially track their movement into the treatment zone, the light source, and document that. And so this is a trial that we conducted. It's going to be a little bit dark, I think, but at 2 minutes and 3 seconds, what you're going to see is a brown marmorated stink bug starting to respond to 400 lux intensity of light. And so you can see its antennae sort of moving. It's coming into the crescent of light and starting to respond. And so here comes the insect. It's responding directly to the light. So in this case, this would be a positive response to a particular light source, and we would end the trial at that point. And interestingly enough, when we look at the data from this study, these are just hot off the press over the last several weeks or so, what we are seeing is that, first of all, there's a differential response. We see a stronger response from males compared with females, and it's increasing as we increase the light intensity as well. And so this is interesting because the males are the sex that produce the aggregation pheromone. Also, males respond more quickly. This is their mean time of detection. So in other words, if a male responds to the light source, it responds very, very quickly. And so if the males are responding to the light source and then perhaps releasing the aggregation pheromone, this is all really interesting. And so in collaboration with our colleagues, we're also trying to identify the aggregation pheromone of this bug. This is work being led by Dr. Aijin Zhang with ARS in Beltsville. And so these are headspace collection containers that have male brown marmorated stink bugs where we are collecting the odors, and Dr. Zhang will analyze those, and he's made a tentative identification of the molecules this insect produces. So all of this can be incorporated into our trap design, and so this year we are using the black pyramid traps to detect them, but we hope that over time we can augment these with the true pheromone to make them more sensitive, and then using light to make them more effective as well, based on their optimal wavelengths and intensities. So that's the monitoring tool. The other is the management strategy. So with this, I want to sort of highlight what we've done to identify ways to help manage this insect. We know that we have tremendous season-long pressure around marmorated stink bug. We know, as the Congressman said, that essentially they continuously move into the plots. And what we learned was that the greatest efficacy from some of our management programs occur when the insect has direct contact with finished wet spray material. However, because they are constantly moving in from the outside, only a small portion of the population contact that material. And also, some of the insecticides that are labeled as excellent against brown marmorated, or against our native species, are not so excellent against brown marmorated, and particularly this idea of knockdown and recovery. This is a small experiment we conducted to show this effect of knockdown and recovery with 2 materials, tombstone, a pyrethroid, and vidate, a carbamate. We treated an area of orchard, recovered the bugs, and followed their mortality for 7 days. And what we found was that if you look at these graphs, it shows 3 sort of states for the bug. Yellow indicates moribund, meaning they're not dead, but they're not capable of moving. Green is alive, and red is dead. So a bug that's considered to be alive can actively move. And what we see over time with something like a pyrethroid is that they have a capacity to recover. And so keeping that in mind, this allowed us to think about how we should evaluate insecticides. And the idea being that the primary threat that is posed by this population is the population that comes in after the spray residue is dry. So in other words, we need to look at residual activity as a means for controlling this insect. So with that, we have been able to screen about 40 different compounds in our laboratory. This information has gone out to extension entomologists to allow them to help make recommendations for the 2011 growing season. And so this is a 3-part trial where, first of all, we look at their response to insecticide residues on treated surfaces, and we do this over about 4.5 hours, followed by direct observations of their capacity for vertical movement after exposure, and then again, we track their mortality for 7 days. So these are photographs of them in the horizontal mobility trial. This is a no-choice experiment. They're introduced to dry residue, allowed to forage for 4.5 hours. We take measurements at increments of 10 minutes at 0, 1.5, 3, and 4.5 hours. And what we found, and I'm just going to run through these quickly, is that In most cases, what we see is an effect. So on the bottom, for example, is a worry or a pyrethroid where we see them beginning to succumb to the material, whereas water on the top we see generally at 0, 1, and 2 hours, they're unaffected. And so with this, we were able to look at their mobility. And so we've looked at carbamates, the organophosphates, organochlorine, and oxidizines, looking at how their horizontal mobility is affected over 4.5 hours. Pyrethroids are interesting. They're instantly stimulated, but then they're incapacitated. And the neonicotinoids as well. We then followed this by this vertical movement bioassay I mentioned. Look at their capacity for vertical movement. You can see many of the compounds, they were no longer able to grip vertical surfaces. Then finally, we tracked mortality, and what we did over 7 days, again, with all of these classes of materials, following those 3 categories, alive, moribund, and dead, for carbamates, organophosphates, pyrethroids, and the neonicotinoids. And so one particular compound, safari, dinotefuran, which is one that's not labeled for tree fruit as of yet, is one that we continued with. So this is our lethality index that Dr. Berg will talk about when he talks about management recommendations. But the bottom line is this is one piece of the story. This is their response to dry residues. There are other studies that are coming forward with finished wet spray material, feeding trials, looking at sublethal effects, and ultimately in the field. But the final piece I'll say is that a Section 18 for dinotefuran for tree fruit is being pursued, and Dr. Berg and the state of Virginia will serve as the lead state. And in order to add information to this, we have done additional trials with various rates of dinotefuran and looked at their mortality over 7 days. We've just finished these trials, given the lethality indexes to Dr. Berg, and so this work will continue. And so I'll just acknowledge my lab group who have been integral in providing and working so hard to generate these data, and I'd like to turn it over to Dr. Chris Berg from Virginia Tech, who will talk about the Section 18 process and management of brown marmorated.